for the introduction. Today I'm going to be talking about osmolality testing. So some of you may be aware of osmolality testing, some of you may not be. But from Advanced Instruments' point of view, it is a real critical process parameter within bioprocessing, and especially within cell and gene therapy. That workflow, that application of osmolality testing has a variety of positions throughout the workflow. So today I'm going to talk about various different applications from upstream, downstream, and into fill finish too. Before I go into the applications, I just wanted to give some background on osmolality itself. So osmolality is a basic concentration measurement. So like it says on screen, osmolality is the total number of solutes in a liquid solution expressed in osmoles of solute particles per kilogram of solvent. So because osmolality is not dependent on volume, it is therefore not affected by a change in the solution's temperature. So something to note here is the difference between osmolality and osmolarity with an R. So osmolarity with an R is looking at solute particles per litre of solvent. And this therefore is then affecting that measurement with a change in solution's temperature. Something to also note is within literature, the two terms, so osmolality and osmolarity, are interchanged within literature by various different scientists. And the way to kind of differentiate between the two is via the units. So you're looking at that milliosmoles per kilogram of water as seen here on screen. So what we're looking at when testing osmolality is the measurement done by freezing point depression. So when a solute is dissolved in that pure solvent, the freezing point of that solution is lowered, so depressed. Advanced Instruments osmometers are doing that measurement based on freezing point depression technology. So regardless of that solution or your drug product, for example, if you're adding the same quantity of that solute each time, technically your osmolality should stay the same. If you're adding different solutes, for example, and there is an error in the quantity that is added, your osmolality is going to change. So you're going to be able to see the difference. And this is why osmometers are most commonly seen within quality control laboratories. Again, something to note is that osmolality cannot characterize your solution. It cannot tell you what is in the solution. It can just tell you the concentration of those solutes in solution. So moving on to the technology within the instrumentation. So first of all, I wanted to show this from the European Pharmacopoeia. So for osmolality testing in the European Pharmacopoeia, they directly state that freezing point depression should be the technology used for osmolality testing. And what is freezing point depression? So here is a graph representing what happens when you are introducing your sample, your solution, your drug product into the instrumentation. So if we follow the points on the graph, you introduce that sample at point 1. The instruments are cooling that sample, so you go past your sample freezing point, so you are therefore supercooling your solution. At point 3, we are initiating freezing. So within our instruments, we are doing this by mechanical agitation. If you have AI's instruments, you will know that by kind of a knocking noise. And what this is doing is it is initiating the nucleation or crystallization of that solution. With this energy release, you can see there is a temperature increase also at point 4. So you have that heat of fusion released. And then finally at point 5, this is where you'll be taking your osmolality measurement. So you're seeing that plateau of temperature, and this is where you're seeing your osmolality result. And like it says, the sample freezing temperature correlates to the number of solutes in solution, and this is what our instruments measure. So today, I really want to go into more detail in terms of applications of osmolality within cell and gene therapy. So within the monoclonal antibody space, SOPs is set. Osmolality can be seen as a critical process parameter within that workflow. But I think the cell and gene therapy space has a lot more potential for osmolality testing currently. So first of all, I wanted to talk about media QC, media quality control. So we know that media is going to be either made in-house or that media is going to be bought from external vendors. But either way, that quality control is extremely important with this solution. But how is osmolality testing going to help here? So that media, as most of you will know, will have to be a specific osmolality specification to ensure appropriate cell growth, cell health, cell viability, and checking the osmolality of this media before it's introduced into the process is really critical. That incorrect osmolality, whether it's too low or too high, could have real detrimental effects on your process moving forward. So like it says here, we've got some references throughout the presentation. So for example, an incorrect osmolality could result in lower growth rate and viable HEC293 cell concentrations, which could then therefore lead to growth inhibition and also ineffective vector production. My point down the bottom is risk mitigation is also key. So an example here being a company that we have spoken with was not osmolality testing. They were hydrating their media in-house, making their own batches, and they could not figure out why their cell cultures were dying. The viable was low. They tested everything. What was happening in this solution? To cut a long story short, basically they found out that the purified water system that they were hydrating their media with was actually contaminated with lead from the system. So those pipes were actually adding lead into their media. They realized this by osmolality testing. Their osmolality was extremely high and therefore again detrimental to their cells. So without that process parameter, they would not have been able to resolve this issue. Moving more into kind of the cell culture expansion, more of an in-process monitoring tool. So again, we need osmolality to really establish cultivation conditions. And this is looking at preserving that cell viability and productivity. We know that everyone is going to be doing a different cell culture process. There's going to be different supplements, different medias, different feeds, and this is all going to impact your osmolality differently. Again, if you're looking at extreme media osmolality results, you are potentially compromising that cell growth rate and viability. So ideally, for your osmolality result, you're looking at potentially between 280 and 320 milliosmoles for your cell culture. However, there is literature now that shows that higher osmolality has been used to slow growth to maximize productivity. There is a lot of literature out there, again, in the MAB workflow space, looking at shifting osmolality to a higher value to then improve MAB titer and productivity. We wanted to do some studies in-house, and we did this alongside Cell and Gene Therapy Catapult, based on the cell and gene therapy workflow. So what we did is we looked at shifting our cell culture to higher osmolalities than that of the control, which is 300 milliosmoles. And we wanted to see at what level we would increase that titer of AAV2. So here you can see different conditions were run. We were looking at shifting the osmolality to both 400 and 500 milliosmoles at different time points post-transfection, so 24 hours and 48 hours post-transfection, with two different agents. So you can see sorbitol and sodium chloride here. It's quite obvious to see that condition 2 was the most successful. So by shifting osmolality to 500 milliosmoles at 48 hours post-transfection with sodium chloride, we were able to increase that specific AAV titer by 25% versus the control. So not only is osmolality an in-process monitoring tool, but it also can be used as an optimization tool as well. Next, I wanted to talk about transfection efficiency. So there isn't as much evidence, as much literature around this application with osmolality, but I still think it's worth talking about. What do we currently know about osmolality and transfection efficiency? So by theory, by characterizing that osmolality, that osmotic pressure within and outside of a packaging cell, it's going to be important to that transfection efficiency and viral yield. I'm not going to spend too much time on this slide, but I just think it's key to note that there is literature out there that is suggesting osmolality as a parameter while looking to improve or optimize the complexing of your DNA and PEI complex, for example, whether you're adding them separately or adding them in situ. Like I said, there is evidence out there, but I think this is an ever-growing application that we're going to see more popularity around in the coming years. Next, viral vector stability. So stability can be seen in multiple ways throughout the workflow. So this specific application is looking at retroviral vector membranes. So here we have 3 different pieces of literature, all focused on osmolality and how you can manipulate osmolality to potentially improve your stability of your retroviral vector. So starting on the left-hand side at the top, you can see that, again, they are using increased media osmolality to improve their vector stability and cell productivity up to a certain limit. So this literature actually was what our collaboration with Cell and Gene Therapy Catapult was based upon. The other 2, again, you see a theme here in terms of increasing that osmolality within the media. So one, you can see that the increased osmolality induced a decrease in the cholesterol to phospholipid ratio, therefore increasing higher stability in the viral vectors that were produced because of this. And again, a similar theme on the bottom piece of literature here. An increase in media osmolality was correlating to retroviral stability. So this is one application of osmolality as a stability indicator, but there are others. So an example being stability throughout that storage and transportation of your drug product. So if you are looking at storing your drug product at a certain temperature for a certain amount of time, osmolality can really be applied pre- and post-storage or transportation to make sure that drug product is safe, stable, and ready to be administered into the patient. So stability, there is a lot of application here, and I think, again, it's an ever-growing application. But viral vector stability, there is a lot of literature currently out there. Aggregation is a real hot topic. I think a lot of people are trying to reduce aggregation or reduce the risk of aggregation. And here, osmolality can really be applied as an in-process check. There is, again, growing literature. Again, it's not one of the most common applications right now, but I think there is a lot of potential for osmolality to be used in this space. We have seen that, again, the MAB workflow is one that has really started to apply osmolality testing to really look into and investigate aggregation, protein aggregation, what higher osmolality could potentially do to aggregation. But this piece of literature is looking at reducing AAV2 vector aggregation via checking the levels of excipients that are going to help reduce that aggregation. So you can see from the table that Wright then looked at screening the different excipients that are known to reduce that aggregation. But what they were doing is testing that level of osmolality to see which was the most optimal osmolality or concentration of that excipient to prevent AAV2 vector aggregation. So you can see it really differs in terms of the different excipients. So you've got a base level of 180 milliosmoles is obviously quite low in comparison to maybe your cell culture, for example. And then it does go up to around 320 milliosmoles. So you're still staying at a relatively low osmolality, but this may differ to what you would normally expect in that solution at this point. Moving into more of kind of the downstream side of things, QC, buffer quality control, is similar to media QC in terms of keeping an eye on your buffers. Are they the correct specifications? Is everything going to be okay before you introduce that buffer into your process? So we actually produced some study in-house here a few years ago, and this was just basing osmolality as a parameter against those usual parameters you would see in downstream. So we did this with pH and conductivity, and I just wanted to show you the data for the conductivity measurements. So on the top row here, you can see some key buffer components and an increase in concentration of those buffer components alongside osmolality readings. You can see that it's quite a linear relationship. You would increase that concentration, and as expected, that osmolality would also increase. If you then compare that to your conductivity, the measurements are all over the place. It's not as sensitive, it's not as reliable, and you really have a difference here in terms of what those increasing concentrations then mean for your conductivity measurement. So osmolality is that direct concentration measurement, whereas conductivity, you can't really say that. We're not proposing that osmolality is an additional, we're not proposing osmolality as a replacement parameter, we're proposing an additional tool. So if you are struggling with conductivity, which we have had conversations with people about, why would you not include a parameter that is quick, that is easy, and is obviously very sensitive to batch deviations or something outside the expected specification? Osmolality can be a quick 90-second test, so it is just something to potentially include into that workflow and again mitigate that risk moving forward. Moving on to more of a newer application. So this data has come from a scientist in the US. So this application is working, there is data being generated, and I think there is a lot of interest potentially around this application for osmolality testing. So within UFDF, this company in the US is using osmolality to check the efficiency of their buffer exchange. So they are measuring the osmolality of their permeate just to see if it is as expected. So they would have known what their osmolality should be at the end of the diafiltration runs. And if that osmolality is not as expected, they know something has gone wrong. In addition to this, what they're actually looking to do is potentially improve their process by looking at the osmolality. So you can see that the osmolality plateaus out from around kind of the 6 diavolumes to the 10 diavolumes. So what they are looking to do is potentially reduce the number of diavolumes in their process based on the osmolality result. So if they are achieving their osmolality result at an earlier point in their process, maybe they can cut down their process with time, by cost, and therefore optimizing their process. There is a lot of interest around this, and I think it is very key to note that you can potentially save your team time and effort by doing this, and again, something that is very easy to implement into your workflow. So finally, the last 2 slides in terms of applications. So cryopreservation, again, a huge topic right now. Everyone is talking about how to improve their cryopreservation process, how to improve their recovery after cryopreservation. So simply, the application here is checking the osmolality of those cryopreservative solutions, and also once included with those cells as well, or that drug product. So testing the osmolality of that cryopreservative before freezing is ensuring that that composition is correct throughout that process. Also measuring post-thaw, again, is just double checking that nothing has happened unexpectedly within that process. There may be contamination, there may be leachables, extractables, something that is unexpected. Osmolality could pick up with such a small deviation within that result. And obviously cryopreservation can really become critical at multiple stages of development, and we are saying osmolality can be used throughout all of them. And finally, actually going back to the cryopreservative formulations themselves. So we have companies working on these formulations, or we have customers wanting to do this formulation internally. And a question that we get quite regularly is, your instruments are based on freezing point depression. These cryopreservatives are anti-freeze agents. Does this actually work? Can they work hand in hand together? And the answer is yes. So you can see that internally we did a study. So you've got some commercially available cryopreservative reagents here. And we tested our specifically designed instrument on these samples. And you can see there's quite a difference in terms of osmolality results. You can go up to over 2,800 milliosmoles with CryoStor CS10. And previously, instrumentation on the market has struggled with these higher concentration samples that are slightly more complex in terms of excipients. But our Osmotec XT was able to do this with minimal coefficient of variation as well. So we know when looking at technology, people have different priorities, but we would kind of consider these 6 things the most important parts of an osmometer selection consideration. Ease of use, so user-friendly. Are all of your users within your laboratory going to be able to get along with this instrument and get up to speed with the instrument quickly? Performance is all about accuracy and precision. Are these instruments going to be able to measure these particularly complex samples within cell and gene therapy accurately and precisely? Volume requirements, obviously within PD, we know sample is precious, so the smaller that volume used for this test, the better, as well as testing time. You don't want to be stood around waiting for hours. This is a very quick and efficient test that we need to get done ASAP. And then the last 2 really work hand in hand. So data management and regulatory compliance. How are you going to manage that data? Where is that data going to go once it's been generated on the instrument? And also, are these compliant with the regulations? Is it pharmacopoeia compliant? Is it 21 CFR? Is it GMP? So we know when talking to customers, this is exactly what they need to prioritize when looking at our instrumentation. So finally, just to kind of run through our portfolio and what we have on offer, we have the Osmotec XT. So I discussed this with the cryopreservation formulation. This instrument has been specifically designed for those higher concentration, higher complexity samples. So this actually goes up to 4,000 milliosmoles, really taking into consideration these reagents that are starting to push those boundaries of osmolality. We have the Osmotec Pro. So this is a semi-automated instrument, loading up to 20 samples at a time, giving that walk-away capability that we know that is needed for a higher throughput laboratory. And then finally, we have our most recently launched instrument, the Osmotec HT. So this is the first 96 well plate-based osmometer to market. HT standing for high throughput here. So again, it's for that laboratories that potentially are looking to do a lot of quality control checks, a lot of screenings, but also within formulation development as well. So just to highlight the different offerings these instruments can give. So we have the large range of osmolality with the Osmotec XT, and all of our instrumentations are pharmacopoeia compliant. So that is EU, US, Japanese, Chinese, all of these instruments are compliant. Fast test time, so dependent on the sample you're testing, you're looking at around 90 to 120 seconds with a small sample volume requirement. So again, for the XT, you're only using 20 microliters of sample. Data compliance, so we have review and approval. We have all of the 21 CFR Part 11 compliance and EU Annex 11, as well as the GMP compliant environments that we can be positioned into. And then finally, the flexible data management. So all instruments can be connected to your network, your LAN, your LIMS. You can use a notebook with them. We have a remote web server. There's also ability to connect to a printer if that is something you're still working with. So just to kind of give an overview here, these osmometers can really be very flexible, no matter if you're working within PD, R&D, or in that GMP manufacturing environment as well. So that is my presentation. Thank you for listening. And everything that I've spoken about today is kind of combined in this application note that you can see on screen, and that is available via our website. We are also at booth 214 if you wanted to discuss any instrumentation questions that you may have. Thank you.
Thanks for the nice talk. Is there any questions? If not, I have some. So my first question is, if your solution is viscous, how is that impact your osmo testing?
Yeah, so viscous solutions in the past have really provided issues for osmometers. So what we can say is that viscosity and osmolality are not linear. You could have a semi-solid gel, but your osmolality is still sitting at 300 milliosmoles, for example. So you're looking at those different molecules and those interactions rather than kind of the concentration adding to your osmolality. So with the XT, we've taken this into consideration, hence why we've extended the range just in case. But what it's more developed for is that increased viscosity of samples as well, that we know we're seeing more frequently, especially in cell and gene therapy, where we're seeing lower dosage volumes in comparison from going kind of IV to subcutaneous, for example.
Yep. My next question is, there's going to be a change in the European Pharmacopoeia Osmo chapter next year. Can you talk us through the changes?
Yes. So with the European Pharmacopoeia in January 24, there will be a change. And there's various different changes within this, but really the biggest difference is within your daily verification check or your accuracy check that is monitored within the EP. So nowadays it's saying that you only would need to do one verification check. So this is what you would run with your instrumentation prior to testing. So we would recommend normally a 290 milliosmol check. However, the European Pharmacopoeia is now changing to include triplicate testing and taking the mean of that osmolality result. So therefore, you're going to have potentially a change in your SOP, moving from one test to 3 for your verification test.
Last question is more on a real application of your instrument. Can your instrument be placed in automated workflow?
The Osmotec HT, so the 96 well plate-based instrument, definitely can be integrated. We're currently working with customers in the US to kind of get robotic arms into them, which it can be done. And it can be placed kind of beside T-cans and automated liquid sealers as well. So there is definitely work ongoing to get the Osmotec HT into these automated workflows.
Great, thanks for your answers.
Thank you very much.